Have you ever wondered why you look different from your siblings? Considering you came from the same parents, it would make sense for you to look exactly the same, right? Well, for some reason, this is not the case, and to understand that, you need to understand meiosis. You might be asking what meiosis is, and that's what me and Aiden are here to explain.
Meiosis is a special cell division of chromosomes when a formation of gametes or reproductive cells occur. Inside one of the two or more gametes, there are 23 chromosomes, and within those chromosomes, there is a gene sequence made up of DNA. But before it even starts, it has to go through interphase. The DNA within the chromosomes self-replicating.
Now, the beginning of meiosis is called prophase. This is when the chromosomes begin to condense and thicken to line up with their homologous pair, which is like the chromosome that has the same size and same type of gene. After they then go into the process of crossing over, which is like when they transfer and exchange genetic information between them. This process also creates recombinant chromosomes, which take the genetic recombination or genetic diversity and variety that siblings have.
Then we move on to the second phase, metaphase, starting with M for middle, because the chromosomes line up in the middle in the sound pairs. In this phase, the spindle fibers also begin to attach to the chromosomes. Then we move to the third phase, anaphase, starting with A, standing for away, because the chromosomes are going to be pulled away from each other. They begin to get pulled away from each other by the spindle fibers that were attached during metaphase.
Then we move on to telophase, with the T standing for two because you have two different nuclei. Soon after, cytokinesis follows, and it splits the cytoplasm. That's not all. We move on to meiosis 2 and the beginning of prophase 2. But prophase 2 is not the same as prophase 1 in the sense that not as much stuff is going to happen. Things like homologous pairs and crossing over doesn't happen. Although the spindle fibers and the chromosomes begin to develop, it's not as eventful as prophase 1. Then in metaphase 2, they still come to the middle, but they're in a single file line, not in pairs like the previous metaphase. In anaphase 2, instead of the chromosomes being pulled away from each other, the chromatids are pulled apart by the spindle fibers. In telophase 2, you're able to see the nuclei reform, and the two cells are each going to divide, and now you have 4 cells remaining.
Then cytokinesis splits the cytoplasm of the cells. Now remember, meiosis, male produces sperm, and meiosis in females creates eggs. Because of the independent assortment and crossing over, there is variety within the offspring of the humans. One example being when a male cell does meiosis, the 4 sperm are different from each other and the original cell they came from because they end up with 23 chromosomes and the starting cells have 46. Because of this variation occurs within siblings because it came from a unique egg and a unique sperm cell, different from everyone that was created.